Good afternoon, everyone. Nowadays we know that the fruits are very rich in micronutrients, and structured carbohydrates. So I have already mentioned that fruits are like a fruit juice are they are contain micronutrients, structured carbohydrates, and the phytochemicals. So nowadays the fruit juices are usually they are treated with the sterilization, pasteurization, and the chemicals. So it is means like, for example, it is affect to the fruit juice quality. So today I brought a new topic called thermosonication. It is nothing but ultrasound with a mild heat treatment. So today I will discuss about this topic called a thermosonication. It influence on various quality properties in the fruit juices. So myself Lakshmi, I'm a PhD scholar from a Department of Post Harvest Management from the College of Kerala Agriculture University from Thrissur. So today I will discuss about what is sonication, types, uses of sonication, what is a thermosonication, what are the mechanism, what are the applications, and some of the related to case study, and what are the advantages and disadvantages, and finally the summary, and finally the conclusion. So normally we will hear the frequency from 20 hertz to 20 kilohertz. So that frequency is known as a normal. That frequency is known as an ultrasound frequency. So we will hear the frequency less than 20 hertz frequency. So if the frequency commonly used to agitate the particle or in a solution, that frequency or that ultrasound frequency, that process is known as a sonication or a ultrasonication. So coming to the what is the ultrasonication? So it is a non-thermal method, and it is a solid liquid gas system used for different purposes, and it is a propagate in a longitudinal way, and during a propagation there is instantaneously pressure variation was observed. And the main principle of the ultrasonication was it reduced the processing time, save the energy, and improve the shelf life of the fruit juice quality, and also maintain the quality of the fruit products. So here they have classified, like Urango one scientist and co-workers, they have classified the ultrasound along into two classification. One is the low energy high frequency in contrast to the high energy low frequency. So in a low energy high frequency is commonly used in the sonography, and high energy low frequency commonly used in the our food products. So this is an sonicator. This is an sonicator, and we call this is an a probe. And when this is known as an a bath sonicator, here the transducers are downside. I will show here. This is a transducer. This is a device which converts the electrical energy into ultrasound energy, because of there is a radiation is absorbed in the juice. And in ultrasonication, in a bar type, the transducer will be in a bottom, and from there it will convert the electrical energy into ultrasound energy. So here the Urango and the co-workers, again, they have observed that after treating the ultrasound processing in the fruit juices, they observed that there is an increase in the quality, shelf life, and also in antioxidant activity, physical stability, and phenolic content. But they observed there is a required in the like less amount of the processing temperature, processing time, enzymatic browning, energy consumption, and the microbial load. Here, some of the uses of the ultrasonication. Ultrasonication was used in the freezing, means nothing but while freezing the food product, there is a formation of a crystal that reduce the formation of a crystal by the ultrasonication, and also used cutting, because this ultrasound commonly used in the non-destructive cutting of the fruits and vegetable, and also used in the drying in order to reduce the in order to means you can call the escape of the moisture from the food product in order to make the dry products like a dry fig, and is also used in homogenization, is nothing but proper mixing of a juice. It is also used in filtration, and is also finally used in the extraction in order to extract the compound from the cell. This ultrasonication was used. So coming to the method of the ultrasonication, there are the many methods are there. Ultrasonication already I mentioned. This method they are using ultrasound along with the only ultrasound, sorry. So in thermosonication, they are using the ultrasound along with the mild heat temperature. So in nanosonication, they are using the thermosonic like a pressure along with the ultrasound. And manothermosonication, in this method they are using like a pressure, temperature, and the like normal ultrasonication. So coming to the main topic called a thermosonication. So here, what is a thermosonication? It is a technique we call that it combine the mild heat temperature from the 30 to 70 degrees Celsius with the ultrasound treatment to enhance the inactivity of the enzymes and microbes in order to increase the shelf life of the fruit juice. So, and is also a potential alternative to the thermal processing technique. So we'll see how it is more like a thermosonication it will work. So when see the big bubble here, when we'll treat the sound waves with the juice, there is a formation of a bubble. So after some time, again there is an increasing in the bubble size. And again after sometimes, if there is an if which means like this bubble was reaches to maximum size, and that would be bombarded with a high pressure and the temperature pressure with the thousand Kelvin and sorry 1000 bar pressure and 500 Kelvin. Because of that, and is also affecting to the neighboring molecule, and is also bubble will be burst within a second. Here you see that this alone sector we call that a sorry, it is an ultrasound. Nothing but it is a probe, and it is connected to a time control system, impulse and power. When this is nothing but ultrasound, it is connected to the thermal, like a thermal, then we can call a thermal temperature control system. The whole setup is known as a thermosonication. So we'll say some of the some of this there are two mechanisms which is involved in the thermosonication in order to increase the shelf life of the fruit juice. In the first one is a microbial inactivation. So we will detailedly I will explain about that how it will involve. So for example, when the like we can call it an apple juice and any juice, when the juice was heated with the or it is treated with the ultrasonication and plus heat is nothing but I already mentioned it is a mild heat treatment. So whole setup is known as a thermosonication. So there is a formation of a bubble because of the high ultrasound waves they leads to the formation of bubble, and again after some time it leads to the increase in the bubble size, and finally it will burst and leads to formation of a free radicals, also known as a water lysis, also known as a sonolysis, that is an H plus and OH minus. So here the mechanical energy plus the free radical which is formed by the bursting of the bubble, that all this combinedly it is affecting to the cell wall of the microbial, that leads to formation of the poration or a cavitation or a pseudoporation. That leads to the that leads to the leakaging of the microbial or a microbial compound from the microbes, and in that means directly it will be affect to the microbial inactivation. So coming to the next mechanism, we call an enzymatic inactivation. How it is involved in the enzymatic inactivation in the juice? Here, before that, we'll see the some of the enzyme which is usually affected to the, or we can call it as a bad affected to the fruit juice. For example, polyphenol oxidase and a peroxidase. This both enzyme which is responsible for the formation of the browning in the juice. And in a pectin methylesterase enzyme which is responsible for the formation of a pectin, but the pectin which is usually it will be an affecting to the fruit juice quality, because nowadays we are preferring that fruit juice should be little bit thickness or little bit consistency should be there. Because of using of pectin methylesterase, then the degrading of the pectin will be absorbed. And ascorbic acid oxide, this is enzyme which is responsible for the destruction of the vitamin C. So because of that we are going for the thermosonication treatment for the fruit juice. So we see how it is involved in the enzymatic inactivation of fruit juice. See, for example, here thermosonication. So when the juice was treated with thermosonication, there is a formation of a bubble, or there is a formation of bubble and is also activate, and then leads to the enzymatic depolymerization. Usually for forming the enzymatic depolymerization, they're involved in the two mechanism. One first mechanism, so first we'll see the first what are they involved in the first mechanism. So when the thermosonication is nothing but the fruit juice is treated with the thermosonication, there is a formation of bubble, and that leads to increase in the size, and finally there is a destruction of the bubble molecule and is also affected to the neighboring molecule. That leads to formation of a cavities in this enzyme, and cavities form usually the formation of the cavities in the enzymes molecule, and is also affected to the degradation of the like what you call primary and the secondary protein structure, and leads to the loss of enzymatic inactivation. So coming to the second mechanism here, due to the formation of a bubble, there is a formation of the free radicals like H plus and OH minus already mentioned before. Because of this enzyme like formation of the free radicals, as we know that the free radicals are they are like very usually it is like affecting, it is a presence of the free radicals means usually it is affecting the body, also is affecting to the oxidation also. So here, like this formation of a free radical due to the due to the thermosonication, and this free radical it will bind to the enzyme substrate in order to inactivate or in order to unavailability of the free site for the receptor site of an enzyme. That leads to the change in the depolymerization of the enzyme molecule and leads to loss of the enzyme activity. So coming to some of the factors which are affecting the thermosonication. First one is the viscosity. See here, for example, if the higher the viscosity, there is a thermosonication needed, it will be more. So in a higher the temperature, if you're using a mild temperature means like 30 to 70 degrees Celsius, that is no thermosonication, because during that temperature or in between that temperature, usually maintain the bioactive components, and that is the main important for the fruit juice. So if you're using the higher the temperature means there is an effect into the like biotic components and everything. So coming to the frequency, like if you're using a lower frequency with a high energy or a high energy, that leads to the like effective of the use of the thermosonication. And it's also sort of called gas bubbles. If the higher the number of the gas bubble formation, higher the poration, higher the formation of free radicals, it is affected to the enzyme, it will also affect the microorganisms. And is also amplitude. If the higher the and the number of the amplitudes are more, that leads to the more poration in the cell juice, that leads to effect on the microorganisms also as an enzymatic inactivation. Next one is the pressure. If you're using a higher pressure, there will be a more formation of the bubbles for the direct bubbles, also for the neighboring bubble also. Another one is time. Here the time is very important. Like here the thermosonication, there like within a fraction of a second there will be formation of bubbles, and it's also breakdown to the neighboring molecules. So with this process, the thermosonication process, it involved in the it required number like 3 to 5 minutes to overall completion of this process. So we'll see what are the applications of using of thermosonication except from the like fruit juice. It's commonly they can use in the dairy industry to mainly for the formation of yeast and fermentation like that. They also used in the wine industry in order to extract the pigments from the cell, and from the cell this is nothing but from the cell wall of that grape wine, and in order to maintain that colority in the in the wine. And also the wastewater treatment, they also used in municipal wastewater treatment in order to remove the particular compound which is responsible for the affecting to the wastewater, or affected to the wastewater. Another one is a pharmaceutical manufacturer. Here they are using a herbal extraction. So here herbal extraction is nothing but they are extracting the compounds like biotic components from this cell. For example, if you're extracting for example, if you're taking the cinnamon, like we can call a cinnamon. So we like cinnamon, for example, we are taking some compound or a herb we can call a madhunashini. If you're extracting, so if you're using a thermosonication or ultrasonication, so there is a more extracting of the what you call more extract of the compound from the cell. It will get more compared to the tradition, because it will break down the cell. That leads to more releasing of the component from the cell into the our extraction. Next one is in a biotechnology. So they're also used in cell disruption in order to collect, or in order to use, or uses the enzyme also to collect the protein component from the cell or from the microorganism also. Next one is in a chemical processing. This is an emulsification link. Emulsification, it is nothing but emulsification is nothing but like mixing of the oil in water. That proper homogenization was mixing was done by the like we can call thermosonication. So coming to the some of the advantages of the thermosonication, one is an homogeneity. I already mentioned. Yeah, there is a greater homogeneity was absorbed, and there is a greater mixing was absorbed, and there is a reduced energy use. And we are also using a temperature from the mild like 30 to 70 degrees Celsius. Because of that, we are maintaining our biotic components in the fruit juice without affecting anything. And so there is an increase in the production. How we can how we can tell how this we can explain, or like we have like for example, if the juice was just by grinding the juice, we will get the very little amount, like we can tell 5 ml. If you're using the ultrasonication or a thermosonication, there is an increasing in the production of the juice, because of the there is a more releasing of the component from the cell. Next one is also maintain the flavor, not affecting any component in the juice, and is also preservation of nutrient was absorbed, and it is also act as an antimicrobial effect, and is also inactivate the enzymes, and is also main thing is it is an alternative to the traditional method. So coming to the disadvantages of thermosonication, here there is an inadequate processing equipment is required, and is also for standardization of a technology for specific fruit juice is required, and is also it is high costly, because here for example, if you're treating an apple juice, for that, we required a different temperature, time, like frequency, volt, and everything. So it is like we have to be standardized to particular juice and particular treatment and particular time, temperature, and everything. This is a critical problem in using of the thermosonication. Except that there is all little retention, it absorb everything. Is was wonderful. So based on that, we'll have taken one case study to explain. One Muhammadin Abid and co-workers, here we see, they have studied thermosonication as a potential quality enhancement technique of an apple juice. So they have studied on the apple juice. So we already have told that this is an ultrasonication. When this is conjuncted with the temperature control system, that whole setup is known as a thermosonication. So we are talking about the thermosonication here, and it is connected to the power pulse time control system. So here they have Mohammed and co-workers, they have took a 80 ml of the apple juice in a jacked vessel, and they have treated the thermosonication with a probe type. This is a probe, so we can set up called a probe thermosonication with a probe type, but is a bar type is there, but they don't mention in this article. Here we see that here the Mohammed and the co-workers, here they have yeah, these are the, like I can tell that this is the table which shows the effect of the treatment parameters for thermosonication of an apple juice. So if the thermosonication was treated with an apple juice, like in a bar type and the probe type, we clearly see in a bar type column, we see that they have treated the juice at 20 degrees Celsius. It is there here at 20 degrees Celsius for 30 minutes and 500 volt, and maintain at 0.06 watt per centimeter cube. And is also in second type, I'm like in a second treatment, they have treated the apple juice in a bar type only at 40 degrees Celsius for 30 minutes, maintain at 500 watt, and radiated at 0.06 watt per centimeter cube. And in third treatment in a bar type, the apple juice was treated at 60 degrees Celsius for 30 minutes and maintained at 500 watt and radiated 0.06 watt per centimeter cube. But in a probe type, they have treated they have treated three treatments. In one treatment, again they are made a treatment one, treatment two. Like an apple juice was treated at 20 degrees Celsius for 5 minutes and 10 minutes of interval, radiated at 500 watt, and maintained at 0.30 watt per centimeter cube. Again, in second set, they have treated the apple juice with the probe type at a 40 degrees Celsius, maintained at 5 and 10 minutes radiation, 10 minutes, and they also radiated 750 watt per centimeter watt and 0.30 watt per centimeter cube. And in third setup, again they have treated the juice at 60 degrees Celsius and maintained at 5-10 minutes like variation, and radiated at 500 watt per centimeter cube for sorry 570 watt, and maintain at 0.30 watt per centimeter cube. So we see the result here. Then after treating the part type and the probe type, this is a part type. TSB is a bath type, and the TSP is a probe type. I already mentioned here. So in a LAB value, this LAB is nothing but is a color which indicates the color of the juice. Like L indicates the dark to light, and A indicates to green to red, and B indicates the blue to yellow. So here, like we see, this is in a control. We'll see that this one is control. In a bar type, among the all the bar type, this is the bar. Among the all the bar type, here the apple juice was treated at 60 degrees Celsius for 30 minutes, is giving a significantly increasing in the LAB value. We clearly observe here compared to the control and compared to the other treatment. So when coming to the probe type, among the all the treatments, the probe at 60 degrees Celsius for 10 minutes, so it's giving increasing in the LAB value compared to the all the other probe type. So however, as a result, like when compare the probe type and the bar type, this probe type is showing the significantly increasing in the LAB value, because here the probe, it is a probe, we already mentioned the probe is like this type of probe. So the probe, it will produce this the, because the probe like for example, I can tell in a transducer there is a length will be more. So probe, it will be very small area. If a more smaller area, the more number of the ultrasound formation will be more. Because of that, that leads to the more number of a bubble, that indirectly it is affecting to the and formation of a bubble, and maximizing the size, and directly it will bursting of the cell, and leads to the homogenization of mixture, that finally affecting to the quality of the juice color. So coming to the this table usually shows the effect of a thermosonication on ascorbic acid, totaphenolics, and flavonoids in an apple juice. Here again, when we see in a control, like in a among they have studied among the ascorbic acid, totaphenolics, and flavonoids. These are the biochemical compound. This for example, in bath type, among the all the bath type, again the bath, like again the fruit juice was treated at 60 degrees Celsius for 30 minutes. It's giving a little bit reduction was absorbed due to the longer the exposure, because of 30 degrees Celsius. So longer the exposure, there is a little bit reduction was little bit, and reduce was absorbed like ascorbic acid, totafenols, and flavonoids. But in probe type, again there is, we see that among the all the probe type, the juice was treated at 60 degrees for 10 minutes. There is an also an absorbed little bit reduction was absorbed in ascorbic acid, totafenolics, and flavonoids. Here this is, there is a little bit reduction was absorbed, mainly due to the we are exposing to the higher temperature here. Here they are exposing 60 degrees for 10 minutes. But however, when we compare the bar type and the probe type having DNA showing the highest significance in absorbed in ascorbic acid, totafenols, and flavonoids compound compared to the bar type. So here they have Mohammed and co-workers, they have finally concluded that the apple juice was a thermosonicator with a probe type at a 60 degrees for 10 minutes at 750 watt, maintained at 0.30 watt per centimeter cube under the 20 kilohertz of the frequency. Means they are absorbed, there is an increase in color value in activation of an enzyme, and also they absorb the better retention of the biotic components like ascorbic acid, totafenols, and flavonoids. So coming to the conclusion of my seminar, here we have concluded, we can came to know that the thermosonication, it is a novel technology, and it is a more energy efficient, and is a potential alternative technique to the conventional heat treatment, and is also enhance the quality and the safety of the food product. And nowadays the demand for the fresh and the higher quality juices with the absence of the additives and preservatives, this thermosonication was using. Coming to the future thrust of using of thermosonication, so there is an optimization of the by changing a time, temperature, frequency, and what you call it is like time and temperature and frequency, and then also in a power for individual juice is a difference. So because of that, we need a optimization for particular juice, and is also we have to there need to be any detailed understanding of the effect of the ultrasound in a combination with the temperature. Thank you.
Thank you very much, Ms. Laxmi, for a wonderful presentation. I will take up the questions now. In the
Okay, ma'am.
meantime, can you please stop sharing the screen so that we can see your full video?
Okay, ma'am. Can I go back, ma'am?
You can stop sharing the screen.
Okay.
So that,
Yeah, ma'am.
yeah, this is fine. Okay.
Yes.
The first question is, how
Yes.
does thermosonication influence the vitamin content in fruit juices compared to traditional pasteurization methods?
Yes, ma'am. In pasteurization method, we are using a higher temperature like 85 degrees Celsius for 30 minutes, especially for the fruit juices. But in thermosonication, so we are using a temperature only for 30 to 70 degrees Celsius. So like many of the biotic components, they are using like many of the biotic components, which is usually it is affecting to the like we can call it as a very sensitive to the temperature. So here in thermosonication, if you are treating also, we are observed the inactivation of enzyme is also observed and inactivation of the microorganisms observed along with the maintenance of the biotic components. But in the pasteurization, we are only the inactivating the microorganism because of the higher temperature. There is a degradation of a like biotic component for absorbed.
Okay. Are
Yeah.
there any specific antioxidants that are better preserved or enhanced through thermosonication?
What man?
Are there any specific antioxidants that are better preserved or enhanced through this process?
Usually, no, ma'am, like thermosonic, like antioxidant, like we can call it vitamin C also. So here, no, usually the 60 degrees Celsius a maximum for the we can call the it is an 60 degrees Celsius maximum. If you're treating the juice at 70 or 80, there will be degradation of a vitamin C because vitamin C only for it will tolerate up to 60 degrees Celsius only, ma'am. You know, on at the 60 degrees Celsius, all the bacteria like antioxidant it will be remain, ma'am, in the juice. All the like antioxidant activity.
Okay.
Yeah, ma'am.
How does thermosonication compare to other non-thermal and thermal pasteurization methods in terms of microbial safety?
Yeah, ma'am. Because here, like in a thermosonication, main important is the ma'am we are using here mild heat treatment. That is the main thing. Because in the pasteurization, we are using higher the temperature. So we are removing or we can degrading the biotic components. And along with another one is in a normal temperature. We are in conventional temperature. We are going for 100 degrees also for like we are maintaining for the so in order to like inactivating of the enzyme or inactivating of the microbe, we are going for 100 degrees Celsius. But we are not focusing on their like biotic components because in thermosonication, why I am preferring that, ma'am? Because in a mild heat temperature also, we are using or we have doing the retention of the bacterial components along with the inactivating of the enzyme and inactivating of the like what you call microorganisms, ma'am.
Okay. How
Yeah.
does thermosonication affect the sensory properties such as taste, aroma, and color of fruit juices?
Yeah, ma'am. Coming to the color here, for example, thermosonication, there is a formation of a bubble, ma'am. That leads to increasing the formation of the higher the size of the bubble, and that leads to breakdown of the bubble. Because of the breakdown, it will affect to the neighboring molecules. It is also affecting, ma'am. Because of that, more number of the bubble formation of juice that leads to the homogenization of the juice. Because of that, there is an increasing in the color value. I already mentioned LAB value was increased compared to the control. In control, there is no homogenization was observed because some of the clumsy was observed in the juice. But when a proper mixing, that is usually done in the thermosonication, ma'am.
Okay. How does thermosonication influence the viscosity and texture of fruit juice?
Viscosity, for example, ma'am, if you are using that one, that is a drawback for the thermosonication because we are using an ultrasound waves. The ultrasound waves, it usually
Hello. Hello.
Hello, ma'am.
Yeah, I will take up that question again. We were talking about how does thermosonification influence the viscosity and texture of the fruit juices?
Yes, ma'am. So, ma'am, like no, ma'am, for the thermosonication, there is a drawback is the main viscosity because the ultrasound in ultrasound there is a formation of an number of waves will be there. If a smooth, like for example, in water or in the air, it will be propagated very easily because there is a no media was thickness was observed, only clear will be there. So, in viscosity, if higher the viscosity will be there, means there is then a thermosonication required a longer period to what you call to what you call inactivating of the enzyme and the microorganism. It required a longer period compared to the lesser viscosity of the juice, ma'am.
Okay, I'll move on to the next question. What is the impact of thermosonication on the shelf life of the fruit juice?
Yeah, ma'am. So for like in many of the articles I have preferred or I have referred that like if you're treating a thermosonication, we can keep for juice for 1 month, ma'am. For example, like normally if you're treating with higher temperature, we can keep in the fridge. For example, in fridge we can keep maximum juice up to 15 days. We have fresh juice. I am asking without adding any preservatives. Because in thermosonication we are not adding any preservative, just only we are giving the thermosonication treatment. So if you are treating a thermosonication, so we can keep up to 1 month. They have preferred some article, ma'am.
Okay, I'll take up the next questions.
Okay, ma'am.
What are the advantages and disadvantages of thermosonication over other technologies?
Yeah, ma'am. Many are the technologies are there, ma'am. Like a supercritical fluid extraction in that treatment, or like man already mentioned manothermosonication. Manothermosonication and thermosonication, somehow it is like similar effect was observed, but in manothermosonication, like three treatments are there, like ultrasound and temperature and the pressure. That treatment is what you call is a more effective compared to the thermosonication. But I have preferred here only thermosonication because here many of the fruit juice is affecting by the biotic components due to we are giving higher temperature because of based on that criteria or based on that what you call area. So I have preferred the thermosonication effect.
Okay. What are the challenges and limitation in scaling up thermosonication process for commercial use?
Yes, ma'am. In thermosonication, so already mentioned this is an advantage. This is a disadvantageous because there is huge. It is an inadequate of the that installment of the thermosonication was required more, ma'am. Like it will be required high. It is also high cost because like, we are using a probe. If your probe is a more effective, but it is when a very we can call it is a more high cost to install that one. But in a bar type, it is a less effective compared to the probe, but it is an easy to install within a commercial also, ma'am. In nowadays, for example, in nowadays, no, ma'am, there's a thermosonication they are using as a pre-treatment. Many of the increasing the shelf life for the fruit juice, not also they are using many of the dairy industry also, ma'am.
Okay. What are the potential future application of thermosonification beyond fruit juices in the food industry?
Yeah, ma'am. So I have mentioned before they are also using in the pharmaceutical for extracting the pigments. How we can also think that how they are extracting. So ma'am, when we treated the ultrasound to not a it is like a herb. We are treating to that one in a flask. We are putting a number of the leaves and we are keeping on the ultrasonication because of the ultrasound waves. There is the wave which will go into the cell wall and release the compound into the which all we can call it solvent. We are using some of the what solvent to extract the compound from the herb. Many of these are like for the pharmaceutical industry or they also using biotechnology because they are mainly focused on the what do you call enzyme extraction or a protein extraction from the microorganism also from the plant, also from the other component also, ma'am. So like they are also using a wastewater treatment also, wine industry also, especially the wine industry, they are using a thermosonication, ma'am. They're giving a ultrasonication with a frequency from 20 hertz to 20 kilohertz. Because of that frequency, there is a releasing of the pigment which is usually present in the cell wall. That leads to the increasing in the increasing in the high consistency of the color in the juice, in the sorry, we can call it wine.
Thank you. Thank you very much. We have now completed the round of questions on behalf of agricultureinformation.com. We like to thank Ms. Laxmi for the talk and for answering the questions. I like to also thank all the participants in the meeting. This meeting will now be closed.
Okay, ma'am. Thank you, ma'am, for giving for my opportunity, ma'am.
You're most welcome.